Protein synthesis reading. Depending on its complexity, an organism may have several hundred to several thousand different proteins as part of its makeup. Each protein molecule is made up of long strands of amino acids. There are 20 different amino acids. A protein may consist of hundreds or thousands of amino acids, and all of these amino acids must be arranged in a particular sequence for the protein to function properly. In fact, all structural and functional characteristics of a protein are determined by its amino acid sequence. DNA stores and transmits the information needed to make proteins in the form of genes. However, DNA does not actually use that information to synthesize proteins. Protein synthesis is the function of RNA, or ribonucleic acid. The process of protein synthesis is accomplished in two stages. The first stage is called transcription, which occurs in the nucleus. The second stage is translation, which occurs in the cytoplasm. The structure of RNA. Like DNA, RNA is a strand of nucleotides. However, RNA differs from DNA in three important ways. First, an RNA molecule consists of only one strand of nucleotides instead of two strands of the DNA molecule. Second, RNA has ribose as its sugar rather than deoxyribose. Finally, RNA has the nitrogen base uracil instead of thymine. In addition, RNA exists in the following three structural forms, and each plays a different role in protein synthesis. Messenger RNA, mRNA, is a single uncoiled strand that transmits information from DNA for use during protein synthesis. It serves as a template or pattern for the assembly of amino acids during protein synthesis. Transfer RNA, tRNA, is a single strand of RNA folded back on itself in hairpin fashion, allowing some complementary bases to pair. tRNA exists in 20 or more varieties, each with the ability to bond to only one specific type of amino acid. Ribosomal RNA, rRNA, which is RNA in globular form, is the major component of ribosomes. Transcription. RNA is produced from DNA through a process called transcription. RNA molecules are transcribed according to the information encoded in the genes of DNA. An enzyme, called RNA polymerase, first binds to a DNA molecule and then causes the separation of the complementary strands of DNA. The enzyme next directs the formation of chemical bonds between the bases of a DNA strand and complementary bases of RNA nucleotides that are floating in the nucleus. RNA polymerase then moves along the section of DNA, establishing the sugar-to-phosphate bonds between the RNA nucleotides in much the same way as in DNA replication. However, when creating the complementary strand, the nucleotide with the nitrogen base uracil replaces thymine. When RNA polymerase reaches the end of the gene, there is a sequence of bases on the DNA that acts as a termination signal. This triggers the enzyme to release the newly made RNA. All three types of RNA are transcribed in this manner. Each type of RNA then leaves the nucleus and travels to the cytoplasm, where it is involved in the synthesis of protein. Notice that the transcription process creates a single strand of RNA with a base sequence complementary to DNA. During transcription, this genetic code of DNA becomes inherent in the sequences of bases in RNA. The genetic code is the system that contains information needed by cells to make structural proteins and enzymes. The genetic code is built into the arrangement of the nitrogen bases in a particular sequence of DNA. Since DNA makes RNA, which makes proteins, The DNA ultimately contains the information needed to put the amino acids together in the proper sequence. During transcription, mRNA is created using DNA as a template. The genetic code inherent in the DNA is rewritten in the sequence of bases in mRNA. Each group of 3 bases of mRNA is called a codon. In effect, each codon codes for a specific amino acid. Each codon attracts a group of bases on tRNA, and each tRNA has a specific amino acid attached to it. The table lists all of the 64 possible codons. Note that each amino acid listed in the table has several codons. These codons differ from each other only by the base in the 3rd position. A few codons do not code for any amino acid. Instead, they are start signals that engage a ribosome to start reading the mRNA molecule, or stop signals that cause the ribosomes to stop reading mRNA. The information in the table is universal. For example, UUU is the codon for the amino acid phenylalanine in bacteria, mice, and humans. AUG is the universal start code. Every organism has the same genetic code. Translation. The process of assembling protein molecules from information encoded in mRNA is called translation. The process of translation begins when mRNA moves out of the nucleus by passing through the nuclear pores. The mRNA then migrates to a group of ribosomes in the cytoplasm. If the protein is going to be used in the cell, it is synthesized by ribosomes floating in the cytoplasm. If it is going to be exported out of the cell, it is synthesized by the ribosomes in the rough endoplasmic reticulum. Amino acids floating freely in the cytoplasm are transported to the ribosomes by tRNA. Notice that each kind of tRNA has a region that bonds to a specific amino acid. Notice also that the opposite loop of the clover-like structure of tRNA bears a sequence of 3 bases called an anticodon. The tRNA anticodon bases are complementary to the pair with the codon of mRNA. This allows it to bind to the mRNA strand. The assembly of a protein begins when the mRNA attaches to a ribosome. The AUG codon on the mRNA pairs with an anticodon UAC on a specific tRNA. Since the tRNA with this anticodon carries methionine, the first amino acid in the chain is always methionine. However, this does not mean that all polypeptides begin with methionine, because this first methionine is later removed from the chain. As a ribosome moves along the strand of mRNA, each codon is sequentially paired with its anticodon, and the specified amino acid is added to the chain. An enzyme in the ribosome catalyzes a reaction that binds each new amino acid to the chain. The process continues with each amino acid being linked to the chain in turn. Eventually, the ribosome reaches a stop codon. This codon brings the process to an end. At this point, the mRNA is released and the protein is complete. References for this reading: Curtis and Barnes, Invitation to Biology, published by Worth Publishers, Incorporated in 1981. Johnson and Raven, Biology, Principles and Explorations, published by Holt, Reinhardt, and Winston in 1996. Tolley, Modern Biology, published by Holt, Reinhardt, and Winston in 1989.